Today, I want to talk about how we select candidate genes from GWAS datasets. GWAS datasets are great for identifying parts of the genome that are linked to our phenotypic trait of interest, but the challenge comes from how to go from SNPs to genes. One way to do this is by looking at what we already know. Which genes do we already know are causal for our traits? I did this with elemental uptake data, also known as ionomic data, for GWAS datasets in these 5 species. I also made random permutations of these datasets by taking the same number of SNPs from our real dataset and randomly scattering them across a respective genome to see what is the chance that any part of the genome would be covered by a known ionomic gene. In this graph, you can see that the known ionomic genes are hitting on something real, since they are covering our real GWAS intervals more often than our random permutations. But when we look at that y-axis, known ionomic genes only account for less than 6% of all GWAS intervals in these 5 species. What's likely happening here is that this comprehensive list of all known ionomic genes is still incomplete. Only using gene annotations to select candidates from a GWAS can leave out many unknown genes that have yet to be characterized. Yet using functional annotations is a popular method because they provide confidence that this gene is a true causal gene. So how do we narrow down our list of GWAS candidates without using prior knowledge, but also provide researchers with confidence that these genes aren't a waste of time to verify and characterize? Well, I want to share one new method that I've been working on, where we use analogous GWAS datasets from multiple species and search for their orthologous genes that overlap with SNPs for whatever trait we're looking at. We first start with the GWAS SNPs, which are these black dots, and the genes, which are these gray bars. For each species, we use a linkage range to determine which genes are linked. And then we filter them by selecting only the linked genes that are orthologous to each other. These orthologous genes are the base of our candidate gene list. From here, we can prioritize the list by their false discovery rate and the number of species represented in an ortholog group to help put the most likely causal genes at the top of the list. At no point does this comparative method use gene annotations, which means unknown genes can be in the list and even be a top candidate. And this is especially helpful for species lacking genome annotations. We found a couple of things from using this comparative approach. The first is being our list size greatly decreases. Using SNPs for the uptake of the micronutrient molybdenum, as an example, I can decrease the size of a gene list by a whole order of magnitude by filtering for only the orthologous genes. The second thing is that we are getting more orthologous candidates than would be expected due to random chance. On the y-axis is our ionomic traits, and for simplicity's sake, I'll only show the results for Arabidopsis. The number of Arabidopsis genes found in our real data set is shown by these bigger green dots, and these blue dots are representing 1,000 random permutations of our real data set. For most of these elemental traits, we are finding more orthologous genes than the 99th percentile of permutations, indicating we are hitting on something that isn't likely to be random. So now I have a short list of conserved causal genes that doesn't leave out unknown genes that I can confidently go forward with verifying and characterizing. Thanks for listening.